Work at the Johns Hopkins University, School of Medicine in Baltimore, in Maryland. A principal investigator of an UH2, UH3 grant that aims to image induce pluripotent stem cells in animal models of amyotrophic lateral sclerosis or Lou Gehrig's disease. I'm a professor of radiology and our expertise is imaging. So we're trying to track cells for future clinical trials using these cells. So the gene editing part is as follows. We are using cells derived from ALS patients which have a defect. A gene is not working properly there. That's called the SOD gene or superoxide dismutase, and these cells are gene-corrected. So we have corrected the gene, so the gene is edited, and these are the cells that we are tracking in live animal models. One of the things we're excited about is that we're exploring quite a new imaging technique. It's called magnetic particle imaging, or MPI. It's quite novel. It only has been done in preclinical models yet. There are no clinical instruments, but using those prototypes, we have seen that we can detect cells with very high sensitivity and specificity in a way that resembles nuclear medicine scans, but the problem there, or the problem, the issue there is that you need radioactivity and special chemicals, radio tracers that we do not need. We can use non-radioactive materials. So yeah, it's a great accomplishment because we were not sure if we could see the cells, if it was sensitive enough, if the machine was working properly. So that's great. It was one of the aims of our grant that we needed to fulfill. Well, the next goal in this project is to use cells that are derived from these iPS cells. So we really use the workhorse, the long-term progenitor cell of that, which is called a motor neuron, to see if we can get similar results with the motor neurons. And the ultimate goal, the next step, is now that we have shown that we can do the imaging, to also show that these cells are therapeutic, so that the disease gets ameliorated, the function of these individuals, these animals, is better. And then ideally, if we can correlate the imaging findings with the therapeutic outcome measures in terms of where do the cells localize, how long do the cells alive, how long are the cells alive, because ideally, to know if the therapy works, it takes a while, right? It takes time. But if you can do imaging and you can predict based on your data that it will work at the end, you know it a lot faster. So it's very convenient to have a predictive marker of eventual therapeutic outcome. So that's the next step and ultimate goal.